As long as you went through the midnight for that.
Thank you.
Hey, hey, hey. Okay. All right. That's the last one. It'll be impractical to hand out others. It'll be impractical to hand out others given that we're shutting down. So that's it for the homework. Yeah. Andrew. Let's hand out. Oh, Andrew has a handout here. I put one on your. I thought I did. Maybe I didn't. That's one from Friday in case you didn't get it. This is today's.
Thank you very much.
It's in my other folder. It's in my other folder I forgot to bring down. I'll get you one after. Okay, folks, it's time to begin. We're missing Livia. Yeah, I know she's here. I think she went to check her stuff in the autoclave. Okay. So you've heard the announcements. I'll just quickly reiterate them. The governor has put Michigan under a lockdown. K through 8 grades are open. High schools are now have to be all distance learning. Colleges have to be distance learning. It'd be nice if they graded us according to cases and how well we've done, in which case we'd be remain open. But alas, the sweeping executive order sweeps us in to the you must close as well. You must close order as well. And so all instruction will be via Zoom by Wednesday. And that means our quizzes, our exams, what have you, will all be on Blackboard. So the exams, I'll get them ready Thursday, and you'll have till Monday to do them. If you forget to do them, don't email me and ask for more time. The answer will be no. Friday to Monday is plenty of time to do a 5-question quiz. If you forget to do it, that's on you. All right? So I don't want to get any emails. Can you extend the time? Because the answer is going to be no. Your final then also will be on Blackboard as well. So I haven't decided what kind of final I'm going to do. I'm still mulling that over, so watch this space. Everybody get the handout from last Friday? If you didn't, it's up here. Jenny, you had a question?
Since it is in place on Wednesday, are we still doing the in-person
Tomorrow, because it doesn't start until midnight of Tuesday. And lab will end before Tuesday, midnight of Tuesday. So, and yes, you can take it.
But by I haven't decided what final I'm going to give. It's still going to be, is it going to be cumulative?
Yeah, yeah. It'll be cumulative final, so. Yeah, except morning section, sleep in.
But if I'm in the afternoon section,
Get there on time.
I'll do the morning section. Because that is really.
If you want to come to the morning section and take the exam, I'm fine with that. Then the date is yours. 9 o'clock. So sleep in, get more sleep, enjoy it. Read the paper, have coffee in your slippers, then come in and take the exam. And then the afternoon section, come at your regular time. You should be well rested by then. And then we'll take the exam, and then that'll be it for the semester, yes?
So are we just emailing you the summary of
Yeah.
the article?
Yeah. Yeah.
I think you talked about before, but is there a grade associated with the ATS?
If you improve, then that will help you. If you fail to improve, then it will not help you. And yes, I have students who fail to improve after a whole semester of biochemistry. Don't ask me how, but it happens. Okay. So the Krebs cycle, while my students were taking their exam, I fiddled around. So here's the Krebs cycle. It's on your handout. So we are right here. We're going to finish it. Then we'll talk about how it's regulated. Then we'll talk about a little shunt that plants use called the glyoxylate cycle that allows them to make to make sugars from fat. Animals can't do that. Plants can because of this bypass of the Krebs cycle called the glyoxylate cycle that lets them do that. We'll look at that today as well. And then if we have time, we'll talk a bit about isocitrate dehydrogenase and why it's become so important as a tumor marker, even as a target in some cases for certain tumors. So let's have a word of prayer then. Let's begin. Father, thank you for this cold day. Watch over us, please. Protect us, our sanity or whatever, as we go back into a partial lockdown. We pray that we get the number of cases down and that we would not have the spike in deaths that seem to be inevitable from the spike in hospitalizations. Thank you for these students and their hard work. Take us to the end of the semester now in Jesus' name. Amen. All right, so the reaction before this, we made a GTP via succinyl coenzyme A synthetase. This takes the energy in this thioester, transfers it to a high energy phosphate. That succinyl phosphate is enzyme bound. High energy phosphate is given to GDP, and you make GTP, and then you also get a free coenzyme A to make succinate, free coenzyme A, and a GTP. This is utilized by succinyl coenzyme A synthetase. And we talked about the mechanism. I won't go through that. Then the next reaction is the only membrane membrane-bound enzyme, membrane-bound catalyzed reaction, that's succinate dehydrogenase, which will take the succinate and will reduce it to fumarate, which is essentially succinate with a double bond, in which case it's not succinated anymore, but you get the picture. So this complex, succinate dehydrogenase, uses flavine to carry the electrons. So you reduce the flavine. This then gets shunted directly to the electron transport chain. So those electrons go straight to oxygen to help make water. So here is part, at least, of the succinate dehydrogenase complex. It's the only enzyme that has been bound. There was a paper, actually, I was a part of, where we took this pathogenic fungus and we isolated enzymes from mitochondria, and we assayed the Krebs cycle enzymes, and we could not find succinate dehydrogenase. And I kept saying, guys, it's membrane bound, just go back to the membrane. No, 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 no. You're full of it. No, no, no. So we assayed and assayed and assayed the mitochondrial matrix. All the Krebs cycle enzymes are found except for succinate dehydrogenase. Guys, it's probably in the membrane prep. It's the cyclohexokinase is binding the outside of mitochondria. It's probably bound to the membrane. No, 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 no. And then it turned out I was right. So nice to be right sometimes. All right, so that flavine gets channeled directly into the electron transport chain. In fact, this is a part of the electron transport chain. And you reduce the flavine and you oxidize succinate to fumarate. Notice that this is a trans molecule, a trans molecule. You have to have a trans molecule because the next enzyme, fumarase, doesn't work unless you have a trans molecule. A trans molecule, this whole thing grinds to a halt. So this is very stereo-specific in that regard. So, yeah.
Is that SAG bound to the membrane?
Yeah, bound to the protein that's bound to the membrane. That protein turns out to be a peripheral membrane protein, so you can strip it off pretty easily. So if we went back to the membrane preps, we would have found that, I'm still not going to let that go. Yeah, it's peripheral membrane. Now, because this compound right here looks a little bit like the substrate, this is a competitive inhibitor for succinate dehydrogenase. This is malonate. Malonate. So malonate has been used to inhibit succinate dehydrogenase in a competitive fashion and characterizes kinetics to some extent. So this shows you how a molecule that looks like the site acts as a competitive inhibitor. So the seventh step now, you're going to add a water across this double bond, and you're going to make malate. Malate. And that is fumarase. Fumarase. Fumarase will produce malate. You add the water across, you get a carbanion transition state, then you eliminate the extra, you take up a proton, and that gives you malate. This is also a stereospecific product. You make L-malate. D-malate is not a substrate for the next enzyme, malate dehydrogenase. You have to make L-malate. So fumarate, fumarase will give you L-malate every time. L-malate. And then the final step is malate dehydrogenase, which oxidizes malate to get you right back to oxaloacetate. And this will reduce the last of your NAD. So this is your third reduced NAD that you will get from this reaction. You'll get it from malate dehydrogenase, get it from isocitrate dehydrogenase, and you get it from the alpha-ketoglutarate dehydrogenase complex. Those are the three enzymes that catalyze actions that will reduce NAD plus 2 NADH plus hydrogen ion. So you make 3 NADH per turn of the Krebs cycle, 1 reduced flavine, and 1 TP. And this is pulled through because as soon as you make the oxaloacetate, it immediately condenses with a acetyl coenzyme A. Immediately. That's how you pull this through, even though this G is quite positive. So L-malate is oxidized to oxaloacetate, and you're back where you started from. So that is the delta G/0, the standard biochemical free energy change of this reaction. But the delta G, without any prescripts, is actually negative because the concentration of oxaloacetate in the mitochondrial membrane ends up being pretty low, thanks to the very generous and robust activity of citrate synthase. Now, if you're not feeding enough acetyl coenzyme A into the mitochondrion, this reaction can't occur. So you're going to stall here if you're not feeding enough acetyl coenzyme A into the reaction to prime the pump, shall we say. So this shows you have a cyclic series of reactions. What you make is what you start with originally. You start with oxaloacetate and you rake it. And in doing so, you oxidize the acetate completely to carbon dioxide and then its high energy electrons, which are carried by electron carriers. And as you keep feeding acetyl coenzyme A molecules into this, it'll keep happening. Doesn't matter where the acetyl coenzyme A comes from. This cycle will keep going. So your book has this really nice picture that's quite glary and difficult to read, at least from where I am. It's probably better. Probably the glare from the windows too, because the sun hits these windows just right this time of day. You can see these 3 sections right here give you your NADH. Succinate dehydrogenase gives you your reduced flavine. So again, for each glucose, you're going to make 2 acetyl coenzyme A's. So you'll have to turn the cycle twice. That means for each glucose, you will make 6 NADHs per turn of the Krebs cycle, 2 reduced flavines, and 2 GTPs, and 4 molecules of CO2. The other 2 CO2s, of course, were produced by the pyruvate dehydrogenase complex. And that also gave you 2 more reduced NADs. That will give you a total, therefore, of 6 plus 2 or 8 NADHs. Then you made 2 from glycolysis. That's 2 more. That's a total of 10 per glucose. Yeah.
Now the PDH complex, because it's so large and it contains multiple copies, does it only do 1 pyruvate molecule at a time?
No, can do multiple pyruvates at a time. Yeah. So if you look at this cycle, and again, you learn this, essentially there's a logic to it. As you work it out, as you draw it, it becomes, I think, easier to try and recognize it. The thing I want you to try and understand is that you stick the acetate on the top of the molecule, and then you're going to oxidize the molecule from the bottom. And so that top, that acetyl coenzyme A, then going to eventually become the molecule that is going to end up down here. So you put it on, and then you break it down from the bottom, and you break it down, and then pretty soon what the acetyl coenzyme A is going to end up being the bit that gets oxidized. So keep track of your carbons. So with each turn, you get 2 CO2s, 1 GTP, but it's equivalently energetically to an ATP, 3 reduced NADs, 3 NADHs, plus hydrogen ions, and 1 reduced flating. And if you want to keep running that cycle, you need to take these reduced electron carriers and you have to reoxidize them. How are you going to do that? You're going to use the electron transport chain to do exactly that. That's essentially what the electron transport chain is for. It's for taking these reduced carriers and reoxidizing them. The fact that you make ATP is a huge bonus, but the big task is to reoxidize those so you can keep running your metabolism. Are there any questions on any steps of the Krebs cycle before we talk about its regulation? If not, then we will go to the Krebs cycle and the glyoxylate bypass and regulation thereof and blah, 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 and all that. Okay, the kinder, gentler side of the Krebs cycle. What do I mean by that? Well, the Krebs cycle makes a lot of metabolites that aren't just for catabolism. The Krebs cycle has reactions that produce metabolites that are essential for anabolism as well. So even those organisms whose metabolisms are fermentative will possess some of the Krebs cycle, sometimes even most of the Krebs cycle. So they may have a Krebs cycle that's incomplete. They may have a Krebs cycle that is missing 1 or 2 steps. But many times they'll have most of the steps of the Krebs cycle because they need those metabolites for biosynthesis. So I'd like to introduce you to the anabolic side of the Krebs cycle. That's what I mean by the kinder, gentler side of the Krebs cycle. Many of you are much too old to remember not George W. Bush, but his father, George H.W. Bush. He came right after Ronald Reagan, who was a very successful president, and he campaigned for a kinder, gentler America. And that became a theme of many comic strips. Number one comic strip, 2 cavemen had their clubs over their shoulders talking about a kinder, gentler cave where we asked permission before we clubbed somebody to death or something like that. Some of the comic strips were quite funny. But that was George W. Bush. He also talked about 1,000 points of light. And I remember the Far Side had this hilarious cartoon about a bat leading a cave saying, we're going to have a giant, kinder, gentler cave with 1,000 points of darkness pointing the way. It was vintage Gary Larson and the Far Side. Okay, anabolic side of the Krebs cycle. So, oxaloacetate is used to make phosphoenol pyruvate, which is used to make glucose. If you want to make oxaloacetate, you have to usually utilize Krebs cycle reactions in order to make it. So oxaloacetate will be withdrawn sometimes for the purposes of gluconeogenesis, or you have Krebs cycle-like reactions that occur in the cytosol. Oxaloacetate is also used to make aspartate, and then aspartate is used to make asparagine. If you look at oxaloacetate, stay in the amino group there, you have aspartic acid. That's how you make aspartic acid. It's 1 step. Very easy. Then it's 1 step essentially to make asparagine. And asparagine is also used for the, aspartate is used for the synthesis of pyrimidines. So you sometimes can withdraw oxaloacetate for not just the synthesis of aspartate or asparagine, but also for pyrimidine biosynthesis in fast-growing cells. So oxaloacetate is a big anabolic metabolite as well. And remember, you can make oxaloacetate from pyruvate via pyruvate carboxylate. Succinyl coenzyme A is hugely important because it's used to make hemes, porphyrins for hemes. So all of your hemoproteins, cytochromes, hemoglobin, myoglobin, all those other types of proteins like that use hemes depend on succinyl coenzyme A. And you only make succinyl coenzyme A in the mitochondrion. So very often, heme production occurs in the mitochondrion consequently, because that's the only place you make succinyl coenzyme A. So this removed from the cycle for biosynthetic purposes, particularly in bone marrow cells and things like that. Alpha-ketoglutarate is used to make glutamate. And then glutamate is a precursor for the synthesis of glutamine, for proline, and then arginine in some cases. Glutamate is also used for the synthesis of purines, So alpha-ketoglutarate gets removed from the Krebs cycle very often for the synthesis of these very important amino acids. Citrate also gets removed for the synthesis of fatty acids. There is a pathway to the synthesis of fatty acids that utilizes citrate. Those of you who stick with me for the next semester will take a look at that pathway, but citrate also is a metabolite that gets removed. And then phosphoenol pyruvate, that is made from oxaloacetate, is used, at least in some organisms, to make these amino acids: serine, glycine, cysteine, phenylalanine, tyrosine, and tryptophan. We actually make cysteine in a different way in our bodies. Glycine is also made in a different way, but serine very often gets made either from glycine, which is mostly what we do in our bodies, but in bacteria, that's precisely how you make these amino acids, is from phosphoenol pyruvate. Once again, you're going to remove oxaloacetate for the synthesis of those amino acids. So because the Krebs cycle has kind of two limbs to it, one that's catabolic, one that's anabolic, it gets called an amphibolic pathway. An amphibolic pathway, it serves two purposes. One, it serves a role in catabolism, breaking things down. Two, it serves a role in anabolic pathways whereby you synthesize molecules. So pathways with this dual character are called amphibolic pathways. And that's why many of your organisms that don't have a complete Krebs cycle will have many, many aspects of it. If they don't have a complete Krebs cycle, the enzyme usually that they're missing is alpha-ketoglutarate dehydrogenase, usually. There are exceptions to that, but that's a statement that you could usually take to the bank. So these amino acids that get made from Krebs cycle intermediates, of course, include aspartate. In bacteria that make their own amino acids, there's a family of amino acids called the aspartate family that includes lysine, isoleucine, asparagine, and threonine. Those are the so-called aspartate family. They're made from aspartate. We, of course, don't make those from aspartate, but many bacteria do. And then, of course, glutamate is made from alpha-ketoglutarate, and then there's a glutamate family, arginine, glutamine, proline, and so on and so forth. Heme synthesis requires succinyl coenzyme A. Hemes are the prosthetic groups of several different important proteins, and then oxaloacetate to phosphoenol pyruvate That's important for glucose synthesis or for the synthesis of other amino acids as well. You can use PEP carboxylase or PEP carboxykinase. PEP carboxylase tends to be used more in plants. Animals tend to use PEP carboxykinase more often than phosphatidylpyruvate carboxylase. And as we said, it's used to make glucose or these amino acids. And then oxaloacetate is used to make these two amino acids, which then are used to make several amino acids, and then also, in the case of aspartate, is used to make pyrimidines. Alpha-ketoglutarate also is used to make glutamate, which is then a contributor to the synthesis of purines. Now, if you are oxidizing fats or glucose or proteins, and you're removing metabolites from the Krebs cycle, the cell now goes, doesn't have that metabolite it just made. So it kind of reminds me of a child who was building a series of dominoes, and then their little sister come over and kick them over, and they would say, "Stop it," essentially. Now the cell could say, "Stop it," but that would not be very useful. Instead, the cell has reactions to replenish those metabolites. We call those reactions anaplerotic reactions. They are replenishing reactions to replenish the removed metabolites so that the cell can keep the Krebs cycle a-rolling, so to speak. And here are these reactions. So there's four main ones, of course. There's pyruvate carboxylase, which will replenish oxaloacetate. This enzyme is very, very prevalent in liver and in kidney. It's found in other tissues, but, excuse me, the highest levels of expression are definitely in liver and kidney. You have some in brain and testes and skeletal muscles, but definitely the highest levels are in liver and brain. Phosphatidylpyruvate carboxykinase, very high levels in heart and skeletal muscle. Also, to a lesser extent, liver, kidney, and brain as well. Phosphatidylpyruvate carboxylase, mainly in higher plants, yeast, and bacteria, not so much in animals. You'll see it in things like shrimp and sponges and things like that. It's not very widely distributed in mammals. Malic enzyme, very widely distributed enzyme, replaces malate. Malate's then used to make oxaloacetate. So you replace the oxaloacetate by replacing the malate. By replacing the malate, you push then the reaction towards the production of oxaloacetate and make the delta G more negative. So this will be used to replace oxaloacetate. So these four enzymes I need you to know. Those are your anaplerotic reactions that replace the metabolites that are removed from the Krebs cycle. Let's look a bit at pyruvate carboxylase. Again, this is an enzyme that is, number one, allosterically activated, acetyl coenzyme A. So this enzyme is used in gluconeogenesis predominantly. It is used to make oxaloacetate, which is then used to make phosphatidylpyruvate. If there is too much acetyl coenzyme A, it says it's building up, Krebs cycle is not working fast enough, and you need to slow it down, or maybe you need to go backwards. So this is an enzyme that's activated by excess acetyl coenzyme A. This is a biotin-containing enzyme, and it catalyzes this reaction right here. In order to activate that biotin, you have to expend an ATP. So you will take pyruvate and bicarbonate, that's essentially CO2, that is soluble, and you make oxaloacetate, you carboxylate it, and you hydrolyze the ATP. So this enzyme has a lysine. The lysine attaches the aliphatic chain of the biotin. ATP is then going to activate the bicarbonate. You make a carboxyphosphate. The carboxyphosphate is essentially made at catalytic site number 1. Catalytic site number 1, notice the biotin's going to be able to swing between the two like George of the Jungle. Okay, you guys are too young for that cartoon.
No, we know.
George of the Jungle. Okay, okay, good. All right. Okay. Well, that cartoon was famous when I was a kid, and I didn't know if it still made its way into.
Tom's been remade. Brendan Fraser was George of the Jungle.
Yeah. It's really stupid. You watch it, you just watch it to laugh. It's kind of like, there's a movie that came out in the 90s called Wayne's World. It was really stupid. You don't watch it for a plot. You don't watch it for a deep story. You just watch it to laugh. Because it's got one gag after another. Because you laugh and you laugh and you laugh. It's pretty funny, but it's really stupid. No. Okay, so this. No. Hey, gang. Concentrate, biotin. This swings back and forth between these two. This is made here, and it's going to be used to activate the biotin. Let's take a look at how that happens. This molecule here, as you can see, will be able to attack the lone electrons right here. This will be withdrawing electrons from the carbon. That carbon will attack here. You'll displace the phosphate, and you're going to get a carboxy biotin. The carboxy biotin now gives you a carboxy biotinyl enzyme. So the ATP was hydrolyzed here at active site 1. This swings over here, gets the CO2 put on it. Now it's going to swing over here and deposit that CO2 on a, what's the substrate? Pyruvate. So here's the pyruvate. As you can see here, the pyruvate is going to be attacked by the CO2 because you've got this stuff going on over here. You got withdrawal of electrons. You're going to attack the CO2 here and carboxylate it. And you're going to make oxaloacetate. You have a pyruvate enolate intermediate here. That's pretty reactive. It's going to attack here, and you get oxaloacetate that's made at the catalytic site number 2. So you make the carboxyphosphate here, you hydrolyze the ATP over here. This then has the CO2 transferred here, it swings over here, George of the Jungle. Pyruvate binds over here. You then get the conversion to the enolate. It then attacks here, and you carboxylate it, and then you release that oxaloacetate product. Boom, there you are. All carboxylases use biotin, all of them. So biotin is a pretty important enzyme. And this is why you should not eat raw eggs. Raw eggs contain in the egg white avidin. Avidin has an extremely high affinity for biotin. It binds biotin. Avidin, by the way, is a small protein. It is a little pocket for biotin. Biotin sits in there and it binds it oh so tightly, far more tightly than your body will ever be able to get, be able to swipe it away. So that avidin will take the biotin and you won't be able to absorb it. Now, if you cook the egg, you denature the avidin and it's fine. Yes, I know in the movie, Rocky, or his train, drank raw eggs. Rocky was an idiot, okay?
But you can drink eggs.
Yes, like you have an eggnog. Yes. Eggnog, in my view, is one of the most disgusting drinks ever created. I detest it.
So, and then you aren't absorbing biochem that causes.
Biotin deficiency, deficiency in bio. Why do you think they add vodka to it, for goodness sakes? Because then it becomes more tolerable. Yeah, yeah, yeah. It's pasteurized, so it's heated a bit, but there's probably still some evidence in there. Oh, okay. Okay. Yes, it's disgusting. And that's why people add alcoholic beverages to eggnog so that it now becomes tolerable. Okay.
Why ruin it?
Well, that's how you make it. You put egg in it. Yes, but they're cooked.
Okay, that's fine. Okay.
They're cooked, and then the thixotropy of the mix ice cream under very high shear, because you add carrageenan usually, which is from algae. That shear force also adds to the nature of the avidin as well. All right, Krebs cycle is very heavily regulated. You'd expect it to be like any other major catabolic pathway. So here are the major sites of control. Isocitrate dehydrogenase is kind of the commitment step. So that is probably the most heavily regulated step in the cycle. This step here is also where you convert alpha-ketoglutarate succinylcoenzyme A. That is also regulated. The steps coming into it are also regulated. Notice this side of the reaction, not very heavily regulated, because once you get past this step, you're committed. It's going to happen. If you block this, you don't have to block this. So the cell tends to regulate at key places. It doesn't tend to over-regulate, unlike our government. So you pick a key spot, you regulate that. You don't. Sorry. Okay. Give me a moment, okay? Right. Okay. So excess ATP, if you're making too much ATP, you're making more ATP than you need, that is going to slow down the catabolic pathways, because the goal, the purpose of those catabolic pathways is to make ATP. If you're making ATP and it's not getting used, you'll start to slow down the Krebs cycle, and you slow it down, first of all, at pyruvate dehydrogenase level. Pyruvate dehydrogenase is pretty sensitive to high levels of ATP. Citrate synthase is too, and so is isocitrate dehydrogenase. Those steps are inhibited by excess ATP. Now, normal concentrations of ATP in the cell won't affect it. Once you creep above those levels of ATP, these enzymes will slow down at high levels. They'll be slowed down to the extent where the Krebs cycle is really not going very hard anymore. Excessive reductant also inhibits the Krebs cycle. So notice that these two steps here both produce NADH. So the pyruvate dehydrogenase complex and its kissing cousin, alpha-ketoglutarate dehydrogenase complexes, both of those are pretty exquisitely sensitive to excessive NADH. Both of those are shut down, and notice it's kind of an end product in this case. They're shut down, so is citrate synthase. That's shut down by excessive NADH as well.
Are they shut down at the same time? Because if you stop pyruvate dehydrogenase complex on both, well, stop it or with the previous slide with ATP, then the whole Krebs cycle would stop. So do you stop them all at the same time? Why would it?
You cannot stop them all at the same time. It depends on the levels. And it depends also locally where NADH is being consumed. So this would be the case if you run oxygen, or it'll be the case that there's damage to the mitochondrion, or it might be the case also if you want to shunt some of that to the cytosol for biosynthesis or something like that. They can, under certain circumstances, usually 5A dehydrogenase complex is slowed down first. Then the Krebs cycle usually starts to grind to a halt later. It depends essentially on how steep of an increase you have in NADH. That's usually what determines it. So the pyruvate dehydrogenase complex is inhibited by acetyl coenzyme A, end product inhibition. It's activated by free coenzyme A. It's activated by NAD. In plants in particular, it's activated by calcium ions. Calcium ion activation is pretty minor. In animal cells, you do get some activation, but it's minor. Not like in plants where calcium really, really activates it. And then it's activated by AMP. So if your energy charge is low or your reductant charge is low, that tends to activate pyruvate dehydrogenase complex. Free coenzyme A substrate, and another substrate, those will tend to activate it, whereas an end product, acetyl coenzyme A or NAD or ATP for that matter, will all inhibit it. High levels of ADP activate isocitrate dehydrogenase and citrate synthase. So ADP can activate them and even relieve the inhibition caused by too much NADH and that sort of thing. Calcium ions will activate these two enzymes, isocitrate dehydrogenase and ketoglutarate dehydrogenase. And then succinyl coenzyme A is going to end product inhibit the alpha ketoglutarate dehydrogenase and citrate synthase. So citrate synthase, interestingly, is inhibited by several other things made by other parts of the Krebs cycle. And then high citrate levels, remember, inhibit phosphofructokinase 1 and also inhibit citrate synthase. The pyruvate dehydrogenase complex is also regulated by phosphorylation. So there are two regulatory proteins associated with that ginormous complex. One is a kinase, PDH kinase. The other is a PDH phosphatase. These will respond to intracellular concentrations of ATP. Or maybe we should say intramitochondrial concentration of ATP. So under conditions where ATP levels are quite high, the kinase will phosphorylate E1. By phosphorylating E1, you inactivate E1, you inactivate PDH complex. At low ATP concentrations, you activate the protein phosphatase. That dephosphorylates E1, and that now activates E1. So the ATP charge plays a very, very key role in the activity of the pyruvate dehydrogenase complex. Very, very important. NADH is a pretty potent inhibitor. This is especially the case in plants. It's also the case in humans, but not nearly as much as you see in plants. Fatty acids will also inhibit PDH complex. So that's the regulatory part of the Krebs cycle. Are there any questions on the Krebs cycle, anything like that? So this hopefully will be a study guide for you as you learn the Krebs cycle by rote. Okay. Now, let's pick on Amanda a second. Now, Amanda is blessed with this condition called diabetes mellitus. If she wants to raise her blood sugar, she can eat glucose or she can eat protein because her body can use the protein to make glucose via gluconeogenesis. However, can't she raise her blood sugar by eating fat? No, she can't. Also, if her blood sugar starts to fall, can her cells degrade and use that to make sugar? No. Animals cannot make sugar from fat. Animals cannot make sugar from fat. The reason for that has to do with the fact that if you break down a fat to acetyl coenzyme A, you can use that to make oxaloacetate, but eventually you're going to remove that oxaloacetate for glucose production. You're removing essentially everything you need to run the Krebs cycle. So you see now that doesn't work. Now plants make seeds, and those seeds often store fat, and they use those oils are then used to make sugar, which is then used to make cell walls, then used to make more plants that elongate out of the soil. So if you take, for example, peanuts, you take a peanut, you can, I don't know if Dr. Baldwin has ever done this. He's got that glorious press he uses. I don't know if you've ever used that in no-chem to make peanut oil out of peanuts, but if you take peanuts and take them out of the shell and give them a good crush, you'll get peanut oil. And if you plant a peanut, the peanut will use the oil as a source of energy. It's very light. You don't have to hydrate it, but plants can make sugar from fat. Why? Because they have a bypass to the Krebs cycle that allows them to do it, called the glyoxylate cycle. And here's how it works. All right. Okay, so yeah, animals cannot make sugar from fat. They can make fat from sugar, unfortunately. You eat too much sugar, you're going to get fat. But they can't make sugar from fat. Plants can. Okay, glyoxylate cycle. So the glyoxylate cycle is just like the Krebs cycle, but simpler. So here's the glyoxylate cycle. Take a look at this. You know that one, you know that one, you know that one. So just two more reactions that are different in order to make the glyoxylate cycle. So let's take a look at these particular reactions. So the first two steps are just like the Krebs cycle. You take oxaloacetate, you take acetyl coenzyme A. The acetyl coenzyme A usually comes from the degradation of fat, of fatty acids. You then make citrate, and that is catalyzed by citrate synthase. Then you rearrange it via a aconitase, and you make isocitrate. Now, once you make isocitrate, you have your first unusual reaction in the glyoxylate cycle, and that is you take isocitrate and degrade it to succinate and glyoxylate via isocitrate lyase. Isocitrate lyase. Here's a reaction catalyzed by isocitrate lyase. You convert isocitrate into, you break it right here essentially. You make isocitrate, and glyoxylate. Isocitrate, citrate, and succinate, sorry, and glyoxylate. Succinate and glyoxylate. Now, the succinate can then be used in the Krebs cycle. That's not a problem. So you're going to take the glyoxylate, you're going to condense it with acetyl coenzyme A to make malate. Malate. So acetyl coenzyme A, again, comes from the degradation of fat. You take glyoxylate, condense it with acetyl coenzyme A, make malate, and then, Bob's your uncle, malate dehydrogenase will then make oxaloacetate, and oxaloacetate can then be used for gluconeogenesis. That's essentially how you're going to make glucose from fat. Succinate feeds into the TCA cycle. You convert it to fumarate, convert it to malate, convert it to oxaloacetate, and you're all ready to go again. And then oxaloacetate gets converted to phosphoenolpyruvate via phosphoenolpyruvate carboxykinase, and then that's used to make sugar via gluconeogenesis.
So, you're saying that the plant has a full Krebs cycle and?
Yep. Yep. And very often, the glyoxylate pathway occurs in a little vesicle called the glyoxysome. So the glyoxysome, the fats are broken down actually in the peroxisome, and the glyoxysome and the peroxisome are usually right next door to each other. Next door also to the mitochondria, because they're going to pass metabolites between each other. You virtually see 3 organelles in a trifoil inside the germinating seed. Okay, so oxaloacetate is a substrate for phosphoenolpyruvate carboxykinase or phosphoenolpyruvate carboxylase that gives you phosphoenolpyruvate. You make sugar from that via gluconeogenesis, blah, blah, blah. All right, so in plants, these unusual enzymes, isocitrate, lyase, and so on and so forth, they are located in a special membrane-bound organelle that is called a glyoxysome. Glyoxysome. And so your plants are going to store their lipid-rich compounds in the seeds. The glyoxylate pathway will then take the products of the fats that are degraded in the seed. Succinate, which is a product of this, gets shipped to the mitochondrion, and it gets converted to malate, and then the malate gets shipped to the cytosol for subsequent conversion into oxaloacetate and for glucose. So here in your plant, you have a lipid body, hence where you store the oil. Here's your glyoxysome, and here's your mitochondria, all next door to each other. And anybody guess what that is? That's a peroxisome. So degrades it, sends it out to the cytosol. Boom, there you are. And this essentially is the pathway that is included from the lipid body to the glyoxysome to the mitochondrion to the cytosol. So it's a multi-organelle sort of feature. Vertebrates don't have the glyoxylate pathway. Now, the citric acid cycle isozymes, like malate dehydrogenase, they come in 2 isozymes, a mitochondrial form and a cytosolic form. Vertebrates, however, lack this cycle. So you get partitioning of the citric acid cycle and the glyoxylate cycle, and this allows for coordinate regulation of these respective cycles. So in your germinating seed, you oxidize cells in the glyoxysome / peroxisome / glyoxysome peroxisome partnership. When you oxidize fatty acids via beta oxidation, you make acetylcoenzyme A. You then shunt that into the glyoxylate cycle. You get succinate. Succinate goes from the glyoxysome to the mitochondria. There, it gets converted to fumarate and then malate gets shipped to the cytosol, where a cytosolic isozyme of malate dehydrogenase converts it to oxaloacetate, which is then converted to phosphanidylpyruvate, which is then the stuff for gluconeogenesis to make hexose sugars for the cell wall. Now, your oxaloacetate, remember, can't be transported from the mitochondrion to the glyoxysomes because you don't have a transporter in the mitochondrial membrane for oxaloacetate. So how does the cell do it? It makes aspartate out of it. And then the aspartate is then transported out, and then it gets converted to oxaloacetate in the glyoxysome, and then you condense it with acetylcoenzyme A, and you begin the whole thing anew. So this is a oxaloacetate aspartate shunt. Oxaloacetate aspartate shunt. Now the crucial branch point between these two pathways is isocitrate. So the conversion of isocitrate is, as you might guess, like most branch points, very tightly regulated. And there is a protein kinase that is involved in the regulation of isocitrate dehydrogenase. When isocitrate dehydrogenase is phosphorylated by the kinase, it's inactivated. And now you will shunt your material to the glyoxylate pathway, which will be used to make glucose. And if you dephosphorylate it, now you're going to shunt that stuff through the Krebs cycle for ATP synthesis and so on and so forth. And I see from the clock we're out of time, so we will finish this on Wednesday on Zoom. And then we'll get into the oxidative phosphorylation pathway. We'll look at electron transport, ATP synthase, and everything else. Yes?
So we can expect the Krebs to be over the Krebs.
Yes. Yeah.
What time is it for you? Tonight or like class?
Midnight.
Oh,